Okay, so here's a quick look at the lecture on calcium ions and the role they have in signaling. So calcium ions are used for to control the activity of many enzymes and cellular processes. And the difference between calcium and other second messengers is that calcium ions aren't made, but they are stored and released, and then stored and released. So it's not like cyclic AMP, which is made from a substrate, or IP3, which is made from a substrate, but calcium ions can't be made or broken down, so they are stored and released. So it's the change in concentration of calcium ions that constitutes the signal. Now, we've already talked about the fact that the movement of calcium ions may be controlled through the inositol phosphate compounds, and there are also other mechanisms that exist to control the movement of calcium ions. Now, how does calcium have its effect in the cell? There are some proteins whose action is mediated by binding to calcium. And one of the key examples of these calcium binding proteins is calmodulin. So calcium ions within the cell can be controlled in two ways. Firstly, they can be pumped out of the cell, and secondly, they can be sequestered within an organelle within the cell. On the plasma membrane, there are calcium pumps which consume energy, and also within the organelles there are some pumps that consume energy and some that don't. Now, cells can sequester calcium in the endoplasmic reticulum, the sarcoplasmic reticulum, muscle tissue, and the mitochondria. One of the signaling molecules that is used to control the levels of calcium is IP3. As we know, IP3 is released from the plasma membrane by the breakdown of phosphatidylinositol bisphosphate, or PIP2. IP3 can bind to IP3 receptors, which may be, an example would be the IP3 receptor in the ER, and it can cause the release of stored calcium into the cytosol. And here's a figure which ties in nicely with some of the prior work we've been looking at. So here we have a receptor, it can be a tyrosine kinase receptor or a G protein coupled receptor, which can activate phospholipase C. Phospholipase C cleaves the inositol phosphates to release IP3, which can bind to an IP3 receptor to allow the flux of calcium ions from intracellular stores, such as the ER or the mitochondria. And then these released calcium ions can then modulate the activity of various kinases, typically through calcium binding proteins such as calmodulin, to cause activation of kinases and thus their effect in the cell. So, as well as storing calcium ions in organelles and pumping them in and out of cells, they can also bind to proteins within the cytosol, which also sequesters the calcium ions. And we're going to look at calmodulin as an example of a calcium binding protein. Now, for a protein to bind calcium, it involves oxygen atoms in the amino acid residues. So typically, glutamate or aspartate residues within the protein accumulate, like the protein might have between 6 and 8 of these amino acid residues in one area of the protein, and that can be a region where calcium ions abound. So people have looked at common features of these calcium ion binding proteins, and they've identified a protein structure referred to as an EF hand. Now the EF hand is made up of two alpha helices, referred to as E and F. The alpha helices are positioned in such a way that the two helices resemble the forefinger and thumb of the right hand. So these are the alpha helices, and between the alpha helices there is a calcium binding pocket, which is where the glutamate and aspartate residues are that can bind the calcium. Other calcium binding proteins are calbindin, calsequestrin, and high affinity calcium binding protein. So what is it in the cell that the calcium is controlling? Well, there's a whole bunch of proteins that we've come across which can be controlled by calcium. So we have calcium dependent protein kinase, we have a calcium dependent protein phosphatase, we have phospholipase C, pyruvate dehydrogenase, we have nitric oxide synthase, protein kinase C, there's adenyl cyclase, and there's a whole range of proteins which can interact with calcium or calcium binding proteins, and this interaction modulates their activity. So calmodulin is involved in the mediation of the calcium signal in signaling pathways, such as the generation of nitric oxide and gene regulation, and also in the control of phosphorylase kinase. So calmodulin is involved in regulating these proteins. Calmodulin is an acidic protein with high capacity to bind calcium. The calmodulin protein contains a bunch of these EF hands, but the actual shape of the calmodulin protein resembles that of a dumbbell. So it's got two globular domains on the on either side of the protein, with a long, flexible region which contains alpha helices. So here's a schematic cartoon of this dumbbell structure. We've got the flexible alpha helices in the center, and these globular domains on the end here. Each globular domain contains two EF hand regions. So here's the two binding pockets from each of the EF hands, and this sort of topology makes up calmodulin. The alpha helices has a distribution of hydrophobic and polar residues on either side of the alpha helices, and binding of the calcium causes a change in structure to expose these hydrophobic residues that can then interact with other proteins and therefore regulate other proteins. So we've been talking about the different calcium pumps that exist in the cell to modulate the calcium ion concentration. Within the plasma membrane, there are two types of pumps which can discharge calcium from the cells. One of these pump types uses ATP as an energy source, and another one of these pump types can use an electrochemical gradient across the membrane to drive the flux of ions. The first pump we described, which is the ATPase pump, a plasma membrane calcium ATPase, uses the energy of hydrolysis of ATP to drive the calcium out of the cell. So it's an energy-dependent process, and we consume a lot of energy regulating our calcium ion concentrations in cells. And here's a schematic of the hydrolysis of ATP and the associated pumping out of calcium ions outside of the cell, and also bringing in some hydrogen ions to compensate. Another pump is the electrochemical gradient pump, and this is a sodium-calcium exchanger, and for every 3 sodiums that come in, 2 calciums are exchanged. So calcium ion concentrations in the cell are released and increase, and then are sequestered. So you get these spikes of calcium ion concentration occurring within the cell. So typically the calcium ion concentration in the cell is very low, and then a spike occurs where it rises suddenly, and then it's subsequently restored to its low levels. So what we have is a cycle of calcium ions increasing and decreasing. In addition to the calcium ion pumps that exist on the plasma membrane, there are also calcium ion pumps in organelles such as the ER and mitochondria that we mentioned just a little while ago. So these pumps, there are ATPase driven pumps, and these are referred to as Ca pumps, referring to smooth endoplasmic reticulum calcium ATPase. It's quite a mouthful. So we have these Ca pumps which are driven by ATP hydrolysis, and we have these pumps that we've already come across, which are the ones that are controlled by IP3 to release calcium from ER stores. And this is just a little schematic showing the hydrolysis of ATP to pump calcium ions into the ER, and also likewise to drive calcium ions into the mitochondria. Now, it turns out that the IP3 receptor, which is the receptor which can release calcium ions from the ER, it's not only is it controlled by IP3, but its activity is also modulated by the actual concentration of calcium. So low levels of calcium accelerate the release of calcium from the ER, but then conversely, high calcium ion concentrations inhibit the release of calcium from the ER. So low levels encourage the release, and then the high levels inhibit the release. So we have this kind of bell curve here, whereas at low concentrations of calcium ions, you get an increasing activity of the pump, whereas at high calcium ion concentrations, you get a decreasing activity of the pump. So at both low and high concentrations of calcium ions, the pump is not very effective, but in this mid-range of calcium ion concentrations, the pump's very effective. So effectively, this causes the spike and then turns off the release of calcium ions from the ER. So not only the ER, but also the mitochondria can take up calcium ions through various transporters. And there was some details in the book about the calcium ion pump in the mitochondria. It has a less of an affinity for calcium ions than the ER does, but still it can accumulate calcium ions in very high concentrations, up to 0.5 millimolar within the mitochondria. And also, high calcium ion concentrations in the mitochondria control the activity of pyruvate dehydrogenase, which is an enzyme that's involved in the metabolism of sugars to produce ATP. So there's some sort of crosstalk there between the generation of ATP and calcium ion concentrations, which is important since some of these pumps for calcium ions are ATP driven. So people have also looked at the calcium ion concentration within cells, and it's not uniformly distributed as one may expect, but there are actually hotspots where calcium ion concentrations is higher in one part of the cytosol than another. So clearly there's a mechanism within the cell to reduce the diffusion of calcium ions through the cellular cytosol. If you look at the change in concentration of calcium ions within the cell, it's been observed that there are oscillations or waves of calcium ion concentration. So effectively you get steep rises and then steep drops in the calcium ion concentration, followed by another steep rise and steep drop, leading to an oscillation in calcium ion concentration. And it's thought that the amplitude and the frequency of the concentration oscillation is what may be important as part of the signaling of calcium. So this is just some measurements of the calcium ion fluctuations in calcium ion concentrations over time, and you can see here that there's a frequency of oscillation, and also there's an amplitude of oscillation. So they also and it's either the amplitude or the frequency or both that is important in cell signaling. And this is just a summary of some of the concepts that are in the textbook. In this case, we have our phospholipase C being driven by a G protein coupled receptor to release IP3 and diacylglycerol. The IP3 can control the release of calcium ions from an internal organelle. Also, calcium can be released from the mitochondria, and calcium can also be pumped in and out of the plasma membrane. So the source of calcium ions within the cell can be from organelle stores or from outside the cell. The fluxes in calcium ion concentration lead to the control of various kinases, typically through calcium binding proteins, and then this can control various effects within the cell. And it's the sharp rises and decreases in calcium ion concentrations that I thought would be important, as it's been observed that there are oscillations in calcium ion concentrations in cells. Okay, I hope that helps.